So in that video tutorial we are going to wrap up everything that we need to know from Koch postulate. So Koch postulate was everything was started when Louis Pasteur along with Robert Koch develops the germ theory of disease. That state a specific the disease was caused by a specific type of microorganism, and basically this germ theory of disease is the base of medical world. Okay, so later Robert Koch in 1876 developed for some experimental procedure to prove that germ theory of disease, and later on the basis of those experimental procedure in 1881 he proposed the Koch postulate that can be used to prove any kind of disease is caused by a specific type of agent. Okay, so those four postulates, as we have discussed in our previous video, that the causative agent must be present in every case of disease and absent in a healthy animal. Although nowadays that is not correctly true, because there are many diseases such as some genetic disorders such as immunological disorders are not due to some causative agent, right? Because they are just a misfunctioning of our own body system. So although on the basis of Koch postulate the whole medical world has established, but nowadays all the four postulates somehow are not true. There are exceptions. So the agent of the disease can be isolated from the diseased animal and can be grown on a pure culture, a population of one organism. But we know, and another phenomenon, the second point is also not true, because we know that there are various type of microorganisms, both virus and bacteria, that cannot be grown in pure culture, right? So the disease can be reproduced by inoculating a portion of pure culture into healthy animal, and the agent of the disease can be re-isolated from the infected people and shown to be same as the originally inoculated pathogen, right? But that is not true, because just think about influenza virus. There are genetic possible chance of genetic reassortment, right? That what we call antigenic shift. So there, if a particular cell is infected by different strain of influenza virus, there can be genetic reassortment and production of new type of influenza virus that ensures pandemic, right? So the fourth point of Koch postulate that state that the agent of disease that you have inoculated in a healthy people, after you can also re-isolate it from that infected people and shown to be the same as you have originally inoculated. It is not always true. So fourth postulate, first postulate is the causative agent must be present in every case of disease. As you can see that scientists have find out causative agent with the help of microscope, right? And now this causative agent can be isolated from the diseased people and can be grown in pure culture, as you can see. And now that causative agent can be reproduced in a healthy animal by simply inoculating a portion of pure culture into the healthy animal. Okay, and as a result healthy animal become diseased, and we can re-isolate the pathogen from the healthy animal. And according to Koch and Robert Koch, the diseased causative agent should be the same as originally inoculated pathogen. That means those pathogen and those pathogen are exactly same. So that is the general Robert Koch postulate. So Koch postulate can be used for various practical approaches. So the postulate 1 that said us that the causative agent is present in every case of disease can be used for direct smear and demonstration. The causative agent can be isolated from the diseased animal and can be grown in pure culture. So obviously it should be used in culture, and the causative agent can be re-inoculated by taking a portion of pure culture into an healthy people to healthy animal to test the disease, that is animal pathogenicity test, that is based on the postulate number 3. And postulate 4, that tells us that the causative agent can be re-isolated from the diseased animal and shown to be the same as originally inoculated pathogen. So here we can develop cellology on the basis of that postulate. So there are various exceptions that I have told you. So some diseases such as tetanus have some unequivocal signs and symptoms. Some diseases such as pneumonia and nephritis may be caused by various pathogens, and some pathogens such as asparagens cause several diseases, and certain pathogens such as HIV only infects humans. And Koch postulates are modified to establish etiologies of disease, that means the basis of disease caused by various viruses and some bacteria which cannot be grown on the artificial media. Okay, so that are the exceptions, right? Because he told us that a specific organism must be present or must be behind a specific disease, but pneumonia, nephritis suggesting us that we are the disease that can infect variety of microbes, that can be infected by variety of microbes, right? And asparagens also tell us that I am the microbes that I can cause several diseases, right? So Koch postulate is not full proof. So that is why Koch postulate need to be updated, and that is the molecular Koch postulate developed by Stanley Falcao in 1988. So Stanley Falcao thought that instead of just saying that a specific disease is caused by a specific type of microorganism, I will say that the gene that present inside the pathogenic microorganism that encode a product who contributes to the disease will be the main culprit. Okay, so molecular Koch postulate are again a set of experimental procedure that must be satisfied to show that a gene. So here he take gene as the main player of causing any kind of pathogenicity. So found in a pathogenic microorganism that contribute to the disease caused by the pathogen. So gene that satisfy molecular Koch postulate, he told them as virulence factor. This is very important term, that genes that caused pathogenicity in the host are called virulence factor. The postulate were formulated by microbiologist Stanley Falcao in 1988 and are based on Koch postulate. So in Robert Koch's original postulate, he told that causative agent is the cause of the disease, right? But Stanley Falcao suggests us that no, not causative agent, but the gene is present inside the causative agent are the main culprit of the disease. Okay, so let's see what was the postulate of Dr. Falcao. Although it is also not 100% true, we are going to see. The phenotype or property under investigation should be associated with pathogenic members of the genus or pathogenic strain of species. Okay, that means a particular phenotype, all kind of disease should show a particular phenotype or particular property. Okay, that suggesting us that yes, that particular disease can occur. Okay, for example, in case of polio, flaccid paralysis occur. Okay, so that can suggest us that paralytic poliomyelitis occurred in that person. Okay, similarly, in case of AIDS, you can see various various opportunistic infection. So you can conclude that as opportunistic infection are occurring, for example, esophageal candidiasis, although our candida is a yeast, that is our normal flora. So as esophageal candidiasis occurring, that means there can be a chance of AIDS, that's suppressing the immune system, right? So property or phenotype will always be present in every cases. And additionally, the gene in question should be found in all pathogenic strain of the genus or species, but the absent from non-pathogenic strain. And again, there always be a gene behind every disease. Okay, so a particular pathogen, suppose A, is causing the disease, it should contain a particular gene that helps A to cause the disease. Similarly, same A I mentioned with the help of small a do not cause the disease because it lacks that particular gene. Okay, that means not the causative agent is the main cause, but inside the causative agent, the gene that is present is the main problem of us. Okay, so specific inactivation of the gene associated with virulence factor should lead to a measurable loss in pathogenicity. Okay, and virulence of the microorganism with the inactivated gene must be less than that of unaltered microorganism in an appropriate animal model. That's simply suggesting us that if somehow by nature that particular gene that are simply involved in causing virulence or pathogenicity become mutated, so the virulence should be lost. Okay, or there will be remarkable depletion in causing pathogenicity by that particular organism. Okay, suppose our COVID-19. So nowadays the strain that are in predominant form are not too much virulent than those that we are seeing in case of 2020 or 2021, right? So that is the thing, that there can be a possible genetic mutation that can cause measurable loss in pathogenicity in virulence. So we can use that particular postulate for curing disease, right? That what gene therapy does nowadays, they simply correct the mutation, or if a normal gene is causing problem, so they will simply mutate the gene so that they cannot cause any problem. Although genetic gene therapy is commonly used for treating genetic disorder, but we can use also to treat disease. Now, virulence of the microorganism with the inactivated gene must be less than that of unaltered microorganism with appropriate animal model. Obviously, because microorganism that possess the inactivated pathogen disease causing gene should be less virulent than unaltered microorganism with appropriate animal model, because they are especially the line is for virus, because remember virus is host specific, right? HIV causes infection to us but not to a tree, but not to a animal, right? Similar thing. Now, reversion of allelic replacement of the mutated reversion or allelic replacement of the mutated gene should lead to a restoration of pathogenicity. In other words, introduction of the gene again into the microbe should restore virulence in the animal model. That means if somehow the mutation is reversed or allelic replacement of the gene occurs in the disease causing gene, there should be restoration of pathogenicity. In other words, if another kind of gene that can cause disease reintroduced into the cell, it can restore the pathogenicity in the animal model. Okay, and that is the concept of pathogenicity island, right? Because pathogenicity island, we have seen that with the help of fudge or with the help of another with the help of conjugation, there can be insertion of pathogenic strain or pathogenic gene inside the non-pathogenic bacteria, right? So by that way, a microbe that was inactivated due to the mutation in disease causing gene can be restore its virulence in the animal model. So in molecular Koch postulate, we have learned three postulates. The first postulate simply tells us that the phenotype of property should be associated with pathogenic members of the genus or pathogenic strains of species. Additionally, the gene in question should be found, Additionally, the gene found in all pathogenic strain of the genus or species should not be present in non-pathogenic strain. Again, specific inactivation of the gene associated with suspected virulence trait should lead to a measurable loss in pathogenicity or virulence. Okay, virulence of the microorganism with the unactivated gene should be less than that of unaltered microorganism in an appropriate animal model. Again, the reversion or allelic replacement of the gene should restore the pathogenicity. In other words, reintroduction of the gene into the microbes should lead to a pathogenicity restoration in animal model. Okay, so that are the postulate that Dr. Falcao proposed. So Dr. Falcao's first postulate simply tell us that there should be a particular phenotype or properties for every disease, right? With the help of which you can find out what kind of disease it could be, and the genes that are responsible for pathogenesis or disease should be present in all pathogenic strains of genus or species, but it should be absent in case of non-pathogenic strain. And specific inactivation, there can be specific inactivation of the gene. Okay, so as a result of that specific inactivation of the gene associated with suspected virulence trait should lead to measurable loss in pathogenicity or virulence, and that inactivated or mutated gene should be reversed, or there can be further allelic replacement occur of the mutated gene that could restore the pathogenicity. In other words, reintroduction of the gene into the microbe should restore virulence in the animal model. So there can be still some problem that remains intact, that there are very pathogenic microorganisms whose pathogenicity testing is depend on a specific host. Okay, for example, in case of HIV, what is going on if we want to detect with the help of animal pathogenicity test, we need a human, and that is what not ethically correct. That is why additionally many pathogen actual working for mechanism is still not developed. Okay, and still not described, I should say, and still not clear. Testing a candidate virulence gene requires a relevant animal model of the disease being examined. Okay, that is the point. And ability to genetically manipulate microorganisms that cause the disease, suitable animal models are lacking for many important human diseases, and additionally many pathogens cannot be manipulated genetically. That I was suggesting you in that point, that specific inactivation of the gene could lead to a measurable loss in pathogenicity. That specific inactivation of the gene associated with suspected virulence trait, right? So we can simply inactivate the gene with the help of genetic manipulation, because nowadays we have recombinant DNA technology, we have gene therapies, right? But there are many pathogenic microorganisms whose genetic manipulation is not possible. Again, the reversion of the allelic replacement of the manipulated gene can occur, so it again facilitates the restoration of pathogenicity, and as a result the animal model become again diseased. So that is the concept regarding molecular Koch postulate, that simply tells us that for every kind of disease there should be a gene present. Okay, it was developed by Stanley Falcao in 1988, and the main skeleton is totally taken from Koch postulate. Okay, so that is all you need to know from Koch postulate. It all started when Louis Pasteur along with Robert Koch developed the germ theory of disease, that tell us that if there will be a specific organism present for specific kind of disease. Later in 1876, Robert Koch proposed experimental procedure, and on the basis of those experimental procedures in 1881 he proposed 4 postulates. But there are various reasons why these 4 postulates has various limitations, And on the basis of those limitations, Stanley Falcao in 1988 developed molecular Koch postulate, and he say that there will be genes that encode a specific product that contribute to pathogenicity, okay, inside the microorganism. So we should look for the gene instead of causative agent, and those genes should not be present in non-pathogenic strain, okay. So that is the concept behind the Koch postulate that you need to know. So I hope this video will be helpful to you, and if it is, please hit the like button, share with your friends, and subscribe to my channel to get more videos like that. So thank you for listening to this class.